Depending on which part of the world you're in, good afternoon, evening, or even night, everyone. My name is Luca Vizioli, and the title of my talk today is fMRI at High Spatial Resolutions, Layers and Columns. I have nothing to disclose.
Over the last 2 to 3 decades, fMRI has become one of the main tools to study the human brain non-invasively. Since the discovery of bold contrast in the early 90s, with the growing availability of ultra-high field scanners and the development of efficient softwares and hardwares, we have been able to acquire ever more precise functional images in both space and time. This has led researchers to claim that we are beginning to bridge the gap between fMRI and invasive electrophysiology and optical imaging. But what benefits do we stand to gain by pushing spatial and temporal limits of fMRI? Is it only statistical power, or can we really access novel insights into the human brain functions and study the mesoscale functional organizations in humans and the functional role of some of the most fundamental units of neural computation, such as layers and columns?
During today's talk, I will aim to touch upon some of the main issues in high spatial resolution fMRI. I will start with a brief history of layers and columns, highlight some of the main challenges of submillimeter fMRIs, including some aspects of preprocessing and some of the main acquisition and analytical strategies. I will conclude by talking about future directions. Next, I'm going to talk about layers and columns, providing a brief history and why should we even study them. The first report of the existence of cortical layers occurred over 100 years ago, when Ramon y Cajal first and Brodmann then observed that the cortical ribbon is characterized by different cell concentrations organized in a layer-like fashion across the depth of the cortex. As shown in these figures, layers are easily visible and distinguishable post-mortem with histological staining. The interest around and the importance of these cortical structures dramatically increased when electrophysiological studies demonstrated a specific response pattern across layers, with feed-forward responses being most prominent in the mid layers, specifically layer 4, and feedback in the outer and inner layers. This functional differentiation is further underpinned by a precise circuitry shared across the whole cortex and beautifully summarized by the schematic representation from Guo et al. The circuitry allows specific prediction in terms of the expected laminar activation profile. About 50 years later, in 1955, Mountcastle et al. Observed that functional specification can be organized in a columnar fashion, that is constant across depths. Such examples of columnar organizations include ocular dominance columns, as shown here on the right, and orientation columns on the left. As a testament of the interest revolving around layers and columns, the number of layer fMRI papers has increased exponentially over the last few years. But what are some of the main challenges of submillimeter fMRI? Next, I'm going to talk about some of the main challenges of submillimeter fMRI. Unlike standard fMRI, submillimeter fMRI is a lot less tolerant to error. Pre-processing steps dealing with subject's motions, co-registration between structural and functional data, and cortical segmentation become crucial. Moreover, the SNR gains that accompany the dramatic increase in field strength are traded for nominal resolution of functional images, putting us back into an SNR-starved regime. Questions like what is the true resolution of our functional images become of paramount importance in understanding the feasibility of studying human mesoscale cortical organization. To this end, we have to differentiate between image nominal resolution, sequence resolution, and functional resolution. While these three concepts are tightly linked, nominal resolution refers to the achievable resolution with which fMRI images can be recorded. Then there is what I refer to as sequence resolution, which takes into account issues like blurring in the face encode direction and the spatial correlation in the noise of the image, and therefore tends to be less precise than its nominal counterpart. And then there is the functional resolution, that will be discussed later, which refers to the spatial precision of the functional maps, and it is perhaps one of the most relevant metrics in assessing the feasibility of imaging layers and columns. Correcting for motion becomes crucial. We can imagine, for example, that a 1 mm head movement can be negligible if your voxel size is around 2 mm isotropic. However, at 0.8 mm isotropic, a 1 mm head movement will offset a given voxel of 2 or even 3 layers. Moreover, the choice of the interpolation algorithm is also crucial in preserving the spatial precision of the images. Every interpolation step dilutes the spatial precision of functional images. As shown in this graph, for example, sinc interpolation leads to a minimal loss in spatial precision relative to cubic interpolation, making the former the desirable choice. Co-registration and segmentation are also of paramount importance. Traditionally, due to the lack of contrast and EPI images, we tend to acquire one or more T1 images that we will use to perform white-gray matter segmentation, as well as parcellation of the cortical ribbon into cortical depths or layers. Segmentation has to be very precise to avoid misrepresentation of the cortical ribbon and its layers. As such, unlike standard fMRI where automatic segmentation algorithms can suffice, for high spatial resolution fMRI, segmentation generally requires considerable manual adjustment to maximize precision. Even so, though, in light of heavy distortion of functional images at high field, and the differences in the extent of the distortions between T1 weighted and EPI images, co-registration tends to be suboptimal. While correcting for distortion can ameliorate the quality of cross-modality co-registration, distortion correction represents an additional interpolation step that may be undesirable in that it will further compromise the functional precision of your data. Recently, van der Zwaag et al., an apology for my pronunciation, proposed a different approach based on distortion-matched T1 weighted EPI, which can maximize the precision of co-registration and in principle even avoid distortion correction at once. As shown in the figure on the left, segmentation performed on distortion-matched T1 weighted EPI leads to considerably better results. Consequently, layer activation is also significantly better. For example, as shown here on the right, the quality of mid-layer activation maps is significantly better when using this approach. Next, we're going to talk about acquisition strategies. Different acquisition protocols hold different properties. The main debate about the optimal sequence choice for imaging layers and columns revolves around the trade-off between sensitivity and specificity. This trade-off for some of the most used acquisition protocols is beautifully summarized in the graph here on the right. While there are many interesting sequences that can be advantageous in specific contexts, like for example 3D gradient echo, due to the limited amount of time, over the next slides I'm going to briefly touch upon some of the main benefits and limitations of some widely used acquisition protocols, and provide examples of their applications to laminar and columnar fMRI. For bold contrast, we will look at 2D GE, 2D SE, and 3D Grace. And for non-bold protocols, we will look at CBV VESO. Next, we're going to talk a little bit about gradient echo versus spin echo and their advantages and disadvantages with regards to functional bold maps. One of the main differences between spin echo and gradient echo data is their sensitivity to large draining vessels. As shown in the middle panel, due to susceptibility artifact, for gradient echo data, large vessels lead to high response amplitude, albeit greatly compromising spatial specificity. While gradient echo data are probably blessed with one of the largest SNR and therefore have high sensitivity, they are also plagued by low specificity. Spin echo data, on the other hand, due to the refocusing pulse, are less sensitive to large vessels, holding higher specificity. However, unlike gradient echo data, spin echo images have significantly lower response amplitude and SNR. As a consequence, even when expecting the largest response amplitude in mid layers, Grain echo data display the typical ramping profile of increasing bold responses as a function of proximity to the glial surface, where a large concentration of S cells is present. This point is nicely summarized by the figure here on the left, from Ganza et al., showing that spin echo data peak in the mid layers, while grain and echo in the outer layers. An important concept for high spatial resolution fMRI is the notion of functional point spread function. In vision, for example, point spread function builds upon retinotopic properties to estimate the precision of the functional contrast implemented. As shown on the top left panel, by exploiting the retinotopic properties of V1, we can estimate the hard edge between two gratings by computing the differential map of the activity elicited by the target, left, and the surround grating, right. We can then look at the activation elicited only by the target grating and superimpose the edge location computed on the differential map. With infinite functional precision, we would expect activation to sharply cease once we reach the edge of the stimulus. The fashion in which bold responses drop off as a function of distance from the estimated retinotopic edge of the stimulus convey information about the precision of the signal. Point spread function for 1 mm grain echo data was elegantly estimated by Shmuel et al., bottom left panel. As summarized by the scatter plots on the right, for 0.8 mm isotropic data, Chemo et al. Estimated the point spread function of grain and echo data to be approximately 0.99 mm and 0.86 mm for spin echo data. Therefore, in terms of specificity, laminar and columnar fMRI would benefit from spin echo acquisition. Both measurements of functional precision estimated for spin echo and grain echo data by CHEMO would indicate that sub-millimeter fMRI possesses enough functional precision to distinguish the activity of at least 3 cortical depths, as the average thickness of the cortex is about 3 mm. As we keep pushing nominal resolutions, it remains to be determined whether image functional precision stands to gain from this additional increment in the nominal resolution of our images. Yaku et al. Provide a beautiful example of application of spin echo acquisition scheme to study mesoscale human cortical organization. They demonstrate the feasibility of mapping ocular dominance columns in humans with 70 spin echo fMRI. By contrast, the same map computed with grain echo data appears significantly less precise. Next, we're going to look at a few examples of columnar organization in humans using 3D grays. 3D grays combines EPI readouts with fast spin echo acquisition schemes by acquiring multiple EPI readouts separated by refocusing pulses. Several secondary phase encoding steps are acquired after a single excitation pulse. Slab selective refocusing pulses are applied perpendicular to the slab selective excitation pulse to limit the required phase encoding steps. As evident in these images, 3D grays possesses higher SNR than 2D spin echo, while preserving spatial specificity. The trade-off here is represented by the limited coverage, blue box. Zimmerman et al. In 2011 used 3D grays to study columnar organization beyond V1. They determined that in MT, axes of motions are organized in a columnar fashion. Similarly, DeMartino et al. Used 3D grays to study the organizing principle of the human auditory cortex. They demonstrated that portions of the Heschel gyrus have a columnar arrangement of frequency preference. The same regions are characterized with deep myelin-related anatomical contrast, in agreement with postmortem definitions of area A1. Non-ball contrast, CBV VESO. VESO is an fMRI method that measures CBV changes based on the differences between longitudinal relaxation times of tissue and blood. As shown by these images, VESO possesses higher spatial specificity than grain echo data. As shown on the left panel, VESO is less sensitive to large draining vessels, represented by a drop in intensity values in the flash reference scan. Consequently, the layer profile of VESO data can be more faithful to its neuronal predictions, peaking in the mid layers for feed-forward signals, while gradient echo responses peak in the outer layers. Finn et al. Exploited the spatial precision of VESO to study laminar response properties in the human dorsolateral prefrontal cortex during working memory tasks. They demonstrated that superficial layers are preferentially active in trials requiring manipulation of information held in working memory, and that deeper layers are preferentially active during the response. Recently, VESO was implemented to study resting state network organization and connectivity at the laminar level, allowing differentiating feedback and feed-forward connectivity patterns for the main resting state networks, and confirming prediction of inter-area circuitry. For example, as shown on the leftmost panel, this study demonstrated that V1 receives top-down feedback in superficial layers from V5, while V5 receives bottom-up input in the middle and deep layers. Analytical strategies. Cognitive computational models. In an attempt to prioritize spatial precision, thus far we've been advocating the advantages of high specificity sequences over high sensitivity ones, like gradient echo. However, as mentioned at the beginning of this talk, submillimeters fMRI operates in a highly SNR-starved regime. As such, the superior SNR of gradient echo data may be desirable in specific contexts. An alternative approach to optimizing the acquisition strategy is that of optimizing the analytical strategy to circumvent some of the limitations of specific sequences, like in this case, the limited precision of gradient echo data. One fruitful strategy can be the implementation of cognitive computational models, whereby computational models, for example based on some cognitive or stimulus dimension, can be used to predict activation within a given layer. The cross-validated prediction accuracy can then be evaluated for competing models across layers to determine which model best conforms to the data. Morel et al. Used this very approach to test the prediction accuracy of two competing models in human auditory cortex. They reported that while both responses in deep and middle layers of the primary auditory cortex are equally well represented by a simple frequency model and a more complex spectrotemporal modulation model, responses in the superficial layer are better represented by the more complex model. This indicates an increase in processing complexity in the superficial layer of the primary auditory cortex, which remain present throughout the cortical depths in non-primary auditory cortex. MVPA. It's generally accepted that multivoxel pattern analysis, or MVPA, and more specifically multivoxel classifier, such as linear support vector machines, can be more sensitive than average bold amplitude univariate analysis. We recently replicated these findings, showing that univariate analyses were incapable of differentiating among submillimeters gradient echo average responses elicited by three different images in V1, while SVM, or support vector machines, were very successful in this very endeavor. Left panel. By examining the breakdown of decoding accuracy as a function of misalignment between the training and the testing portion of the data, we further showed that SVM, or support vector machine, can be sensitive to fine-grained structure in multivoxel pattern of activity in gradient echo data. As such, MVPA can be a candidate analytical strategy to extract layer information contained in multivoxel patterns of gradient echo bold responses. We implemented SVM analysis to submillimeters gradient echo data to show that feed-forward activity peaks in the middle layers of V1, while feedback contextual information in the outermost layers. Left panel. We further corroborated these results using a simple linear encoding model to attest that the largest proportion of voxel carrying feedback contextual information resides in the outer layers of V1. Right panels. Vascular models. Another elegant and fruitful analytical strategy is the implementation of vascular models, like those put forward by the Uludaks group here on the left and by the Norris group on the right. This approach is based on the idea that if we understand the flow of blood from one layer to another, we can, in principle, use this knowledge to deconvolve the gradient echo signal to reflect its neuronal rather than its vascular component. The current slide shows an example of blood flow-based spatial deconvolution. The left panel portrays the typical ramping profile of layer responses obtainable with gradient echo data. The right panel shows that by implementing spatial deconvolution and removing the contribution of ascending vein, we can retrieve the expected neuronal laminar profile for feed-forward response, peaking in the mid layers. Future directions. At the beginning of this talk, I argued that with the advent of ultra-high-field scanner, and by pushing spatial and temporal limits of fMRI, we have begun to bridge the gap with invasive electrophysiology. This graph, however, does not accurately depict the current states of affairs. In fact, we tend to optimize one dimension, either spatial or temporal, at the detriment of the other. Recent work, however, like for example that put forward by Lewis et al. PNS 2016, has demonstrated that there are benefits to gain by pushing temporal aspects of fMRI. Consequently, a number of groups have begun to focus their effort to simultaneously optimizing spatial and temporal resolution of fMRI. For example, we recently showed that using gradient echo data with a limited field of view, we can obtain simultaneously spatial and temporal high-resolution fMRI, in this case 0.85 isotropic voxels with a TR of 625 milliseconds. We've also shown that this data possess high enough SNR to compute single subject statistics and reconstruct time courses independently per layer. Along the same line, a recent development of CBV VESO, Magic VESO, is capable of achieving simultaneously spatio-temporal high-resolution fMRI with a spatial resolution of 0.8 isotropic voxels and a TR of 650 milliseconds. Another impressive and extremely promising effort in concomitantly pushing spatial and temporal aspects of fMRI is line scanning put forward by Tyler Morgan and the Muckle group. While greatly limited in the extent of its coverage, using line scanning we can simultaneously achieve 200 micron spatial and 100 millisecond temporal resolutions. By leveraging both space and time, these spatiotemporal approaches have the potential to reveal directionality of layer connectivity, further widening the horizon of human neuroscience. We began this talk by asking what benefits do we stand to gain by pushing spatial and temporal limits of fMRI, whether it was only statistical power or whether we could gain novel insights into human brain functions, and study the human brain at the mesoscale level. There are certainly an outstanding number of important challenges in imaging layers and columns with fMRI, including concerns on whether the available functional spatial precision and SNR and analytical tools suffice to succeed in this endeavor. However, the results are promising and seem to support the feasibility of studying the human brain at the mesoscale level. Before leaving you, I want to show you a little teaser. While still in its optimization stages, and therefore not exploiting yet its full potential, I'm extremely excited to show you the first ever sub-millimeter 10.5 Tesla fMRI activation map. And with this, I would like to thank Prof. Yaku and Prof. Ugur for their continued guidance and support, and of course, thank you all for your attention.